Good evening, everybody. I'm Andrew Creek. I'm a Citrus Development Officer with the New South Wales Department of Primary Industries. I'm co-hosting this event with Bronwyn Walsh from WA Citrus. Tonight, this is an interactive webinar, and it's an output from a Citrus IPDM project that Bronwyn is a colleague with, and it's actually funded by the Hort Innovation using the Citrus R&D funds from the Australian government. Look, we welcome the participation we have from differing citrus regions around Australia to tonight's Zoom meeting or webinar. In southern citrus production, I think an application of autumn copper, it's very important to help stop rind blemish and diseases at winter during harvest. So this evening, we've arranged Dr. Nerida Donovan, our citrus pathologist from New South Wales DPI, and also Rory Tomlinson from Tanuki. They've both prepared informative presentations that will address many of the questions that I get as a horticulturalist, commonly from growers. As I mentioned before, it's an interactive webinar this evening. After each speaker, we will have a question time or a question session. To ask a question, you may raise your hand with the raise hand function. We also have a chat function operating in this evening's Zoom meeting. Questions or comment can be posted in this chat function at any time, and that'll be monitored by myself and Bronwyn. I welcome Nerida Donovan, our citrus pathologist with New South Wales DPI. Nerida is going to share her screen and her presentation. Nerida is going to be talking about citrus diseases that we experience here in the south and also rind blemishes. Please, we'll hold questions till the end to our Q&A session. Thank you, Nerida.
Thank you very much for participating in this webinar. My name is Nerida Donovan. As Andrew Creek said, I'm a citrus pathologist with the New South Wales Department of Primary Industries. I'm actually based in southwest Sydney, in Camden, that's surrounded by an inland sea right at the moment. We're totally flooded out. But I do travel around to the citrus growing regions. So I've been asked to present on diseases and rind blemish, but with a context of the southern growing regions, mostly the inland southern growing regions, and copper sprays. So I won't be speaking much about copper sprays. I'll leave that for Rory. You need 3 factors for disease to develop: a pathogen, a susceptible host, in this case it's different citrus varieties, and the right environment. And if you get all those 3 factors overlapping, then disease can develop. But also under certain conditions, organisms that do not normally cause disease and do not normally behave like pathogens, they can also cause disease symptoms, such as invasion of wounded tissues. So that's why we call them secondary invaders or opportunistic organisms. So they're taking advantage of the situation. So just keep this in mind as we discuss the different pathogens, and we'll revisit this diagram later. I'm not going to mention everything that can be managed by copper sprays, or all the diseases that you would see in higher rainfall areas. So we're focused on the majority. Phytophthora Brown Rot on fruit. The brown rot starts as a slight discoloration of the surface, and then it will spread with the rind turning brown. And in damp weather, you can see like a white fungal growth start to develop. And often blue mold and green mold can come in afterwards. The fruit stays quite firm, though. It's different to some rots that aren't so firm. On leaves, the brown rot appears as a dark brown circular spreading blotches. Greasy spot, which we won't see the fruit symptom. We don't see the fruit symptom in Australia because we've got different species of fungi to what they have overseas. But you can see these sort of raised, sometimes shiny, sometimes not shiny, but that's how they got the name greasy, because they can look shiny if you look at them closely. Irregular lesions. You don't see them often. We used to see it when we had overhead irrigation because of the damp weather needed, but now you only really see it in wet seasons like what we've been having. Anthracnose. Now, this is an example where it can be a bit controversial. Some people say that it's a pathogen in its own right, the fungus that causes anthracnose. But generally what we find is it's a secondary invader. The fungus invades stressed tissue, say after a sunburn, hail, frost, after degreening. So it can occur in the field or it can be post-harvest. And you'll see firm, it can be quite deep brown to black spots on the fruit, and there can be twig dieback. So it can happen. You can see things, evidence of the fungus causing issues throughout the canopy. And Septoria Spot. So this is the last one we'll speak about. So on fruit, it appears first as small, round depressions that over time can expand and join together. They can coalesce and become dark brown, reddish brown to black. You can sometimes get tiny fruiting bodies. They almost look like tiny poppy seeds inside the lesions. So if you've got a hand lens, you could see that. Washington navel and grapefruit are more effective than Valencia, Septoria Spot. Late summer, autumn, you get the infection, and then late winter, spring, the symptoms can appear. But if you spray before the autumn rains, that can stop that process in its tracks. Other things that I just had to mention, even though we're speaking about copper sprays, I had to mention pruning. I know it's a cost, but you can remove the dead wood where a lot of these fungi are hanging around. It improves the airflow, reduces the humidity, reduces the spore load, and reduces the risk of splash. So that's how the spores can move around. Some fungi can have airborne spores, but others can only move around in the splash from either irrigation or rain. Just make sure when you're pruning that you remove the debris from tools and sterilise the pruning tools between blocks. With bleach, that's nothing to do with the fungi. The bleach is because of viruses and viroids that could be spreading around. I would recommend after dipping that you clean your tools again with something else so that the bleach doesn't wreck your tools. So copper sprays, we recommend pre-harvest fungicides. So a copper fungicide spray in autumn, a second spray if you've got a late harvest, and more sprays in wet seasons. So that'll become really clear. I'll talk a little bit about it, but become really clear after the next talk. So successful disease management does depend on even coverage and good retention of copper over all the plant surfaces because it is a protectant. So it will kill the spores and protect the fruit surface where the copper particles are. And there's a using copper sprays to control diseases in citrus prime fact that you can find on the net. There are seasonal differences. So over time, copper coverage declines due to leaf and fruit growth. So as it grows, their areas become unprotected. The action of rain and wind can actually dislodge the copper particles, and more frequent copper sprays are needed if in a high rainfall area, if the season has a higher than normal rainfall, and where overhead irrigation is used. So even if you don't use overhead normally, like as your normal irrigation, sometimes people use it to just flush out the root zone. And so just be aware if you've used copper before that. So we're back to this diagram. So in a typical year, the environment will not be, it'll be drier and the environment would not be conducive. The fungi won't be so happy and won't be able to cause disease. But in a wet year, the environment, the fungi are loving it. They like that damp weather. And so you could have more issues, and that's where we recommend more sprays.
Thank you, Nerida, for that. We're going to have a Q&A session here with Nerida for 5 minutes. Now, Nerida, you touched on the disease Greasy Spot. Michael, I think he was from Sunraysia. He asked what was the best preventative measures for citrus Greasy Spot?
Well, in a dry season where you don't use overheads, you really, the environment is preventing Greasy Spot. But you can apply that copper fungicide sprays. So for Phytophthora and Septoria, it's an autumn copper spray. For Greasy Spot, in a wetter season, you would need to apply copper in the spring and in the autumn. And Greasy Spot is much worse, much more severe where the trees are stressed or there's some sort of other issue going on. And so just maintaining good tree health, which you're all trying to do with best practice management anyway, will reduce your issues with Greasy Spot. Pruning can also, yes, it reduces the number of fungi in the tree canopy, but it can also improve that airflow. So that reduces the issue. It creates an environment that makes the fungi less happy.
Now, I have one more question, Nerida, if I may. Look, we get anthracnose on our fruit in the winter at harvest. Like, where are all these spores living through the summer and the spring because it's so hot and dry?
Yeah, the fungus that is associated with anthracnose can live throughout the canopy. So it can be in the dead wood. It can be so there are some fungi that are only in the dead wood, or in the leaf litter below the trees. But the anthracnose fungi can actually live throughout the canopy. They can be on the twigs, the leaves, on the fruit rind. Often a fruit will have the fungal spores, so the Colototricum, that's the fungal spores on the rind, and actually they will invade the rind, and you just won't see symptoms unless something happens that makes the rind vulnerable and the cells start to die, and then the fungus sort of takes hold and is opportunistic. So basically, what I often say if I get diagnostic samples and I have to plate out samples, you could put an orange next to an agar plate and get Colototricum. Like, you will always isolate Colototricum from citrus because it's ubiquitous, it's everywhere.
From Marianne in WA, what has more effect, actual rain or is high humidity for days just as bad?
It's humidity doesn't help, but mostly the fungi need free water. So that free water could be in the form of rain, irrigation, or dew. So I'm not an expert in meteorology, and I'm not sure how much the actual humidity turns into free water on the leaf. But just think free water. If there's free water on the leaf, then there's the ability for a fungal spore to germinate. And different fungi need different numbers of hours. So it's not even the amount of rainfall. It's the hours of leaf wetness or the hours of free water or wetness on the fruit rind that's important, because different fungi need different numbers of hours in order for their spores to germinate. So it's different depending on what disease you're speaking about. Thank
you, Nerida. I'd like to introduce Rory Tomlinson. Rory's a regional sales manager with Tanuki. He's prepared quite a technical presentation, but I understood it very well actually through his practice run. And look, this presentation will really assist us to best use our copper fungicides. Look, thank you, Rory. You've got the stage, mate.
Well, thank you, Andrew, and thanks to everyone for joining the webinar today. This is my first time doing this, so I'm a bit nervous, but please bear with me. As Andrew said, I work for Tanuki, and you'll see our logo down the bottom here, bottom left-hand corner, and we supply Nordox red copper. So just to start off with, there were a couple of people there who asked about what the best copper product is. Well, I hope this will become apparent during the course of this conversation as to what you should be looking at in copper products. Just to recap on the 5 basic types of formulations, the cuprous oxide, which is red copper, copper oxychloride is green, hydroxide, and I think tribasic copper sulfate are both blue. And then, of course, you've got the copper ammonium complexes, which are the liquid blue aqueous complexes that some of you will be familiar with. So how does copper work? Now, the first thing to keep in mind is that copper products are protectants. They're not systemic products. So they need to be on before the conditions are suitable for disease to get going or to develop. And that's very important, obviously. Once you've sprayed copper, it really won't spread to a large extent across the leaf, and it's very important that subsequent sprays are made, as we will hear a bit later, as the crop grows. Again, as Nerida mentioned earlier in her speech, diseases need free water, so that's important too. keep in mind that copper is most effective on those diseases that need that free water. How exactly does copper work on the leaf? Now, in this schematic diagram here, this is the leaf surface, the green. These are the cuticular and waxy layers. These are plant cells over here. This is, in this case, a red copper particle and a water droplet. What happens is these cells give off what are known as plant exudates. And these are organic compounds that are given out by the tissue, move through the cuticle and the waxy layers onto the surface. And these plant exudates then combine with copper. They help to release the copper from the copper particles in the first place, and they form a complex. That is, the plant exudates with the copper form a complex. There's some kind of equilibrium between free copper particles and those copper particles that are complexed. And the vast majority of the copper particles are complex, as you'll see shortly. So this is a bit of work that was done by some scientists where they looked at how much copper was dissolved on the plant surface, and they looked at beans and citrus, and they found that on beans about 10% of the copper was dissolved, and in citrus it was 25%. And then they investigated further and found that of that 10% that was dissolved on the bean surfaces, 99% of it was complex, and in the citrus 99.9%. So even more was complex, in other words, on the citrus in this case. And what was left in free solution was 100 parts per billion of copper. And that is the same as 0.1 ppm, or parts per million. And many of you will know parts per million as a concentration measure. So in other words, it's very small. And this is the copper that is required to do the job of controlling the diseases. So what affects copper product efficacy? Now, some of you would have heard of Professor Pete Timmer from Florida. He's a well-known citrus pathologist. I think he's retired now. And what he said is that there were two main factors that affect the efficacy of a copper product. First is the particle size, and the second is the frequency of application. So why is particle size important? In the first place, particles can be dislodged by wind. And if they are greater than 3 to 4 microns, wind is able to displace them. The other important thing is rain fastness. Rain can displace copper, as you will appreciate, and you can get direct dislodgement by raindrops of copper particles. And you can also have the copper dissolving in the presence of the water and running off. The second important point regarding the copper product efficacy, as I mentioned, was the frequency of application. Because obviously, as the crop, fruits grow, there's growth dilution that takes place. And for example, in lemons, from the time that the fruit forms to the time you pick, there's about a 14 times increase in the surface area of the fruit. So that needs to be covered over time to ensure that the fruit is protected from disease. The other thing about the importance of frequency of application is that there can be physical dislodgement of particles. Particle size here, we said, is very important. If we look at this schematic over here, there are 2 leaves, and these are 1 micron particles on the left-hand side, and this is a 3 micron particle. The total volume of all these particles is equivalent to 27, which is the same volume as the single particle. And the surface area of the small 1 micron particles is 162 versus 54. So in other words, it is 3 times the surface area of the compared to the larger 3 micron particle. As you can see, you have far better coverage in the first place because of these small particle sizes, and also because the surface area is much larger. This makes it easier for the plant exudates to dissolve the copper on the leaf surface. I mentioned rain fastness. This graph here is based on some results that were obtained with cabbages. Cabbage leaves, as you probably know, are very waxy, and things can run off quite easily as a result. This work was done in France, and they compared red copper with blue and green. On this axis, the y-axis, is the percentage of the copper that was retained, in other words, that stayed on the plant surface. And over here is the millimeters of rain along this axis. So you'll notice that after about 5 millimeters of rain, there was a huge drop in the percentage of copper retained of the blue and green. And of course, it was much higher with the red. After about 25, the red had dropped to about 60%, and the blue and green had leveled off at 20%. Now, the reason for this is simply because the red copper had smaller particle size than the blue and the green. What about solubility of different copper products? Well, effectively, the hydroxides, or the blue coppers, are more soluble. Some of the blue coppers, I should say, are more soluble than the greens, which in turn are more soluble than tribasic coppers, and cuprous oxide, or red copper, is the least soluble of the coppers. And you'll see shortly why that is an important issue. This data, which we have for Nordox, which is obviously our product, and please ignore this small graph, just keep your eyes on the big rectangle here. This is the amount of copper in solution on the y-axis, and over on the x-axis here we have pH. You'll notice that beneath a pH of 6, there is a large increase in the amount of free copper in solution. And so our recommendation to growers always is to keep the pH at 6.0 or above. And if you have a pH meter or pH strips, you can test that from time to time to make sure that that is in fact the case. Between 6 and 7 is fine. All right, so here we have a poll, and the question is, have you experienced any copper spray damage? So if you wouldn't mind answering that question, please, in the poll.
Poll's closing in 3, 2, 1.
Thank you for that. So well, I'm very encouraged to see that the vast majority of you have not had any copper spray damage as far as you're aware. Let's have a look what copper damage looks like on citrus. This picture is courtesy of New South Wales DPI from the prime fact sheet. You'll notice this stippling effect, this black stippling effect. It looks quite similar to melanose, and that is classic copper phyto. And this is just another picture off the internet which shows a variation of that theme. So if you believe you've had copper phyto on your fruit, this is what it should look like. Otherwise, it is probably something else.
Thank you for the informative presentation there, Rory. Bronwyn, we were chatting before everyone was on, and it was interesting. You said you had a lot of wind in WA. You had a question about wind blemish, did you?
Yes, so as you've said, we get wind blemish here and other places of Australia as well. And so does copper make the wind blemish marks more prominent, Rory? And is there a particular way of managing the sprays that can help reduce that effect?
Yeah, thanks for the question, Bronwyn. My understanding is that some copper products can cause blackening of the wind blemish areas, and the good news is that red coppers are less severe in this area, and I do believe from the Sunraysia, for example, that I hear from there that it may result in higher pack-outs because there's less blackening of the wind blemish from red coppers.
Thank you, Rory. Look, is there any further questions before we move on to the next items on our agenda from the floor? Kevin Cock, I think, from Sunraysia has his hands raised.
How's that? There we go.
Yep, perfect. Thank you, Kevin.
My question, I put it in the chat, but I don't think it got through. High volume as opposed to low volume sprayer. We use red copper in the low volume, but I'm always cautious about the strength that I'm putting on through a low volume sprayer and overdoing it. So if you got any clues on what strengths you can put through a low volume, and then I've got to monitor speed and application per hectare as opposed to good coverage and not good coverage. Any clues?
Well, once again, Kevin, please always refer to the registration requirements in terms of rates, the spray volume, and also the recommendation in terms of whether to use high or low volume sprays. I can only say what I said to Joe a while earlier there in response to Joe's question. Is coverage is critical, as Pete Timmer mentioned, and you would need to evaluate your own crop to see if in fact following spraying you're getting what you believe is good enough coverage with a lower volume spray. Certainly, I think up this part of the world, where I live in Brisbane, up here in sort of Gander, Mandubra area, I think people tend to use more high volume sprays. Is the impression I have.
Thank you, Rory, and thank you everyone for those questions and keeping the discussion flowing. Tonight's presentations we're proud to have brought them to you as part of an integrated pest and disease management project that is funded by Hort Innovation and also your citrus levy. Look, our presenters this evening, look, on behalf of the growers, Dr. Nerida Donovan, we thank you for your time and expertise, and also Rory from Tanuki for taking time to prepare those presentations and just answer questions on the spot about differing copper products and stuff. Our co-host this evening was Bronwyn Walsh from WA Citrus. I appreciate your assistance and the technical support pulling this together this evening. I also want to just draw fact that we have published a copper prime fact. It's on the New South Wales DPI website. That draws a close to tonight's evening. Have you got any further comments, Bronwyn, as a co-host and project collaborator on tonight?
No, I think you've done very well, Andrew. For the first time.
That's not one. But look, on behalf of all participants tonight, Nerida and Rory, look, thank you very much for helping bring us all together. And I'm sure if any growers had further questions, they could email you those questions. Thank you, everyone, for your participation. Thank you. Goodbye, everybody. Thank you. Good evening, everybody. I'm Andrew Creek. I'm a Citrus Development Officer with the New South Wales Department of Primary Industries. I'm co-hosting this event with Bronwyn Walsh from WA Citrus. Tonight, this is an interactive webinar, and it's an output from a citrus IPDM project that Bronwyn is a colleague with, and it's actually funded by the Hort Innovation using the citrus R&D funds from the Australian government. Look, we welcome the participation we have from differing citrus regions around Australia to tonight's Zoom meeting or webinar. In southern citrus production, I think an application of autumn copper, it's very important to help stop rind blemish and diseases at winter during harvest. So this evening, we've arranged Dr. Nerida Donovan, our citrus pathologist from New South Wales DPI, and also Rory Tomlinson from Tanuki. They've both prepared informative presentations that will address many of the questions that I get as a horticulturalist, commonly from growers. As I mentioned before, it's an interactive webinar this evening. After each speaker, we will have a question time or a question session. To ask a question, you may raise your hand with the raise hand function. We also have a chat function operating in this evening's Zoom meeting. Questions or comment can be posted in this chat function at any time, and that'll be monitored by myself and Bronwyn. Good evening, everybody. I'm Andrew Creek. I'm a Citrus Development Officer with the New South Wales Department of Primary Industries. I'm co-hosting this event with Bronwyn Walsh from WA Citrus. Tonight, this is an interactive webinar, and it's an output from a citrus IPDM project that Bronwyn is a colleague with, and it's actually funded by the Hort Innovation using the citrus R&D funds from the Australian government. Look, we welcome the participation we have from differing citrus regions around Australia to tonight's Zoom meeting or webinar. In southern citrus production, I think an application of autumn copper, it's very important to help stop rind blemish and diseases at winter during harvest. So this evening, we've ranged Dr. Nerida Donovan, our citrus pathologist from New South Wales DPI, and also Rory Tomlinson from Tanuki. They've both prepared informative presentations that will address many of the questions that I get as a horticulturalist, commonly from growers. As I mentioned before, it's an interactive webinar this evening. After each speaker, we will have a question time or a question session. To ask a question, you may raise your hand with the raise hand function. We also have a chat function operating in this evening's Zoom meeting. Questions or comment can be posted in this chat function at any time, and that'll be monitored by myself and Bronwyn.